to introduce your students?
Yes, I would. Well, I'd like to introduce Alex Partridge. She started working with me a little bit last fall. We started working a little bit on this project here, and she was interested in doing some undergraduate research. And if you're here earlier, you've seen me work with frogs. I do a lot of stuff with that. And yesterday we did some of that stuff, so you hear this again. And earlier yesterday, you heard a talk from Annie Slayton about these invasive snail eggs. And in a talk with her, and actually a trip with her collecting snail eggs, we actually talked about the fact that these snail eggs have a toxin in it. She said, oh, wouldn't it be cool to do that? But her project's so big, she couldn't add into that. Alex was looking for an undergraduate project and said, hey, we got these snail eggs. And she got, hey, this is kind of cool. So we kind of started with that. So I'm going to let Alex tell you about her project.
Hi, well, good afternoon, everyone. My name is Alex Partridge, and today I'm going to be presenting my research on the effects of apple snag egg extracts on the development of the African claw frog embryos. What is the apple snail? They're a freshwater invasive species found in temperate, tropical, and subtropical regions. They're known for their bubblegum pink egg casing. They deposit their eggs inches above the waterline, and they have this pink color that serves as a warning for predators not to eat them. Their eggs contain a toxin that can have neurological effects and even cause fatality. As you can see in these pictures, they are a rather large snail, and they're one of the largest freshwater snails. They about fit in a whole palm of your hand. The apple snails are affecting us here right in Alabama in our ecosystems. They are invading lakes in Mobile, Alabama. They are believed to be released by the aquarium pet trade. A lot of people think they're really cool and want them in their aquariums. However, they outgrow the aquariums and they abandon them in the wild where they overpopulated. They're also destroying the habitats and competing with the natural animals that are already in the area for the resources. And they also carry harmful diseases and parasites such as nematodes and trematodes. You can see in this picture the bubblegum pink casing and the apple snail laying its eggs right above the water. So why did I choose the Xenopus lathus, or the African claw frog, as my model organism? Well, they're very easy to maintain. They're aquatic frog, and they're perfect for the aquariums we here have at JSU. They also produce eggs in large quantities and can be bred year-round using the HCG hormone. This is really beneficial because we can very regularly breed these frogs and get eggs to do experiments on. They also have a short and simple developmental stage of life within the first 96 hours. This helps us really observe the morphological effects on these frogs. My hypothesis was that the apple snail eggs contain a toxin that will adversely affect Xenopus embryo development. My objective was to first find a range where these eggs were being affected from the egg extract, and as well look at the potential toxicity by looking at the mortality and malformation data. The eggs were collected from Langan Park in Mobile, Alabama, and then they were froze until further use for data. In my first experiment, I measured out some of the egg mass, and I homologized it with some phthalate solution. And then the mixture was centrifuged for 5 minutes at 900 RPM. After my first experiment, I realized that these solution had a lot of debris I want to get out. So for the next few experiments, I just changed it where I homologized it and then centrifuged for 10 minutes at 1000 RPM. And then here you can see the centrifuge that I use, as well as the egg extract under the microscope. So the frog embryos were collected. We inject 2 adult frogs with HCG hormone before breeding. They're put in a bottomless chamber. This allows the eggs to fall through and makes it very easy to collect them. When you have the eggs, they have a jelly coat on them that you need to remove. We use 2% L-cysteine to remove this coat. The eggs were then sorted under a dissecting scope. You can see that the bad eggs are usually brown or tan in coloration, while the good eggs are brown or tan in coloration, while the bad eggs are these white eggs. And that's a good rule of thumb to go by to tell which ones are bad and can't be used for experiments. So for my test methods, I would have 4 groups of controls that were kept in phthalate solution. And then for each experimental concentration, I would have 2 groups. Each group or petri dish would have 10 embryos. They were stored in the incubator for 24 degrees Celsius. Every 24 hours, they were removed, and I would take out the phthalate solution, put in another 8 ml, and then re-expose the ones that had concentrations that we were testing. And then every 96 hours, the surviving embryos, control and experimental, were taken out, and they were scored for malformations and mortality rates calculated. For the statistical analysis, we used the SciStat statistical software. This helped me find my mean, my standard deviation. With the preliminary data I have, I was able to find an LC50, but right now I'm not able to do an LC50 until I can get more on malformations, which I'm going to talk about more in a little bit. On my first trial, it was more of a rangefinder. I was very optimistic on the amount of concentration I felt like would kill the embryos. So in this first one, all the control groups had survived, and there was a 100% fatality in the experimental concentrations. And then the second group, it was again a rangefinder. I lowered all the concentrations, but still found a 100% fatality in all experimental groups. Then with the control group, there was a 5% fatality, but that is expected. Sometimes just one in this dish will die and that will affect others, so that's normal. In these pictures, you can kind of see how we can tell whether they're alive or dead. In this picture, you can see where the eggs disintegrated and start to break apart. Obviously, they're not alive. And then this was the control group where the embryos were alive and started to develop. In the third trial, I finally found a range where they weren't dying, so that's good. With a concentration of 62.5 micrograms per liter, there was ones that had survived. And so what I did for the next couple of days is I did a comparison of the mortality rate of the 62.5 micrograms per liter groups and compared it to the mortality of the control groups. As you can see that obviously this egg extract was having an effect on the embryos because the mortality rate of the 62.5 micrograms per liter was a lot higher than mortality of the control group. So for my fourth test, I expanded the amount of experimental concentrations I was doing so that I could find a better range of where they were being affected. So there was, again, some death in the control groups, but that is expected. And then with 60 micrograms per liter and below concentrations, we found a range where they were dying, but not all of them were dying. So we could try to find, exactly the amount that would take to affect them. And then this is a graph of my data. As you can see, as the concentration increased, then the percent of mortality also increased. However, as concentration increased, there was not an increase in malformations. So we don't have any data right now concluding that this toxin is causing malformations. However, we can conclude that it is causing an increase in mortality. Right here, you don't see any data for malformations. This is just because they all died, 100% fatality, so we really couldn't calculate malformations. So these are examples of some of the malformations you may see. In this top right one you have, or top left, this one you have a tail malformation. Right here you have two adenia malformations. On this bottom one, this is one from my control group, and this top is from the experimental group. The experimental group, you can see it has a tail malformation, it has a stunted growth malformation, as well as adenia. So in the results, I did end up finding a concentration range where they were affected. And then the results did support my hypothesis that the apple snail egg extract contained a toxin that will adversely affect the xenopus development. Here are my results from the LC50, my value of 12.181, upper of 18.252, and lower of 6.361. Because I didn't have many malformations yet, I wasn't able to do an EC50, but that's something I'd like to do in the future. So again, for my future work, I'd like to do that EC50. I would like to repeat this data a few times, just so that I can see if my further data would be consistent with what I currently have. I'd like to do a protein analysis. With this egg extract, we're not really sure if it's the toxin in the egg extract that's killing these embryos, or if it's the high protein in these. I would like to do a length growth data, and as well as extend my research to studying effects on different embryos, specifically zebrafish embryos. For acknowledgements, I'd like to thank the JSU Biology Department, of course, my mentor, Dr. Rayburn, and then JSU graduate student Amy Slayton for collecting these apple snail eggs from Mobile, Alabama. Thank you, and does anyone have any questions?